Good evening, everyone, and welcome to the Real Science Exchange, the podcast where leading scientists and industry professionals meet over a few drinks to discuss the latest ideas and trends in animal nutrition. Hi, I'm Scott Sorrell. I'll be your host here tonight at the Real Science Exchange, and we're back with my favorite podcast, which is our Journal Club with Dr. Bill Weiss, where we cuss and discuss the latest in dairy research. First, let's welcome back Dr. Bill Weiss to the table. Bill, welcome back, and what's in your glass tonight?
Welcome again, Scott. Tonight I've got a 50 West with the complicated name called Pilsner.
Pilsner. Now, what's a 50 West? Is that a local brew
local,
pub you've got there?
It's a local brewery. 50 West is used to be a major highway, so they're on Highway 50 West.
All right, good deal. Well, I see that you've invited a guest researcher to the pub tonight. Would you mind introducing him for us?
Okay, the guest today is Dr. Arshad. He's from University of Wisconsin, but I hear he's now in Switzerland. So I don't know if he's updated or not. He'll have to tell us why he's in Switzerland. But this is a paper he did, I think, at Wisconsin. Are you a postdoc or on faculty at Wisconsin?
No, I was a postdoc at Wisconsin.
Okay. And it's a meta-analysis. It's called Effect of Feeding Rumen-protected lysine during the postpartum period on performance and amino acid profiles in dairy cows. And I'll let you discuss some of the co-authors before we get into name the co-authors before we get into the paper. But welcome to the podcast.
Yeah, welcome, Dr. Arshad. And this is not your first time to the pub. I think you've been here once, maybe twice before, as a student spotlight at the ADSA. Is that right?
Yes, it is correct. I guess when we met at ADSA in Missouri, we recorded 2 podcasts there on choline. So it's not my first time. You're right.
Yeah, good. So welcome back.
Thank you.
Now, in the theme...
Hey, I want to add also, it's not your first time publishing a meta-analysis, correct?
Yeah, it's. I think it's now 4th in this field.
Is that
so
right? Yeah, good deal. So, kind of in the theme of the Real Science Exchange, I understand that you're originally from Pakistan. So if you had something in your glass from Pakistan in the pub, what kind of a drink would that be?
it will be milk, and you put soda in the milk. So this is the very common drink we used to drink in summer.
Oh, well, that's interesting.
Yeah.
Well, I like the milk part, that's for sure.
Yeah.
All right. And I don't want to forget my trusty, rusty co-host, Dr. Clay Zimmerman. Clay, welcome back. Good to see you again, sir.
Yep. Thanks, Scott. So what's in your glass tonight?
Clay, and I've had this before, but I'm having a Rock Valley Spirits single malt whiskey. It's a barley scotch. I didn't know I was a scotch drinker, but I'm really enjoying this one. It's quite tasty. So it's made very close to here. It's made by the Milk family, which I found kind of interesting. Maybe that's one reason I kind of like it. So that's what I'm having tonight. And before we get started, gentlemen, here's to a great podcast. Cheers.
Cheers.
The economics of feeding Reassure precision release choline. Reassure is fed during the transition period, and because it's fed for such a short period of time, it costs just $15 per cow, and yet the benefits will continue to generate income throughout the year. Cows fed Reassure produce 5 pounds more colostrum, which pays for your Reassure investment on the very first day of lactation. Cows fed Reassure also produce 5 pounds more milk per day, every day. That means after the first day, every day is payday. Invest in Reassure during the transition period and recoup your investment on the very first day of lactation. After that, you got it, payday. All right, Bill. To begin, can you just kind of give us an overview of why you selected this paper?
Well, like amino acids are big now, and the NASEM really emphasized amino acids. So I've learned a lot on amino acids in the last couple of years. This was just out in 2024, so it's new. And it's a meta-analysis, and that's kind of good if you don't know if you haven't been reading the research for the last 20 years or so. This kind of summarizes it, but then it also quantifies the relationships between it. So it's a good way to get caught up on old literature plus new results from that old literature.
And has there been a lot of research, Bill, on lysine in dairy cattle?
Well, he cited, or the authors have listed 43 publications. I actually have a question for Usman. You have 43 pubs, but you ended up only using 10. And I think the inclusion criteria is important in meta-analysis. So can you tell us why you ended up with 10 publications out of that pretty big list of pubs?
Yeah, no, that's a good point. So, the way I learned to do meta-analysis from Dr. Santos is that the meta-analysis should be hypothesis-driven. And since there is already huge variability, you are collecting data from different worlds, from different regions where people do these studies. There is already quite a variability in studies. Every study with a different setup of diet, with a different setup of environment. So you try to reduce the minimum. You try to reduce the variance whatsoever you can during designing the inclusion criteria. So the reason why we have only 12 studies and 13 experiments, because the inclusion criteria quite a rigid one, because we wanted to enroll only those experiments where people supplemented lysine in a rumen-protected form. Many of those studies infused lysine either in the abomasum or in the intestine, but that is a whole different scenario, right? Because the idea was to see what happens when you supplement a product which is encapsulated in the diet on the performance of these cows. So that was one of the major criteria. The second criteria which I choose, and this is my personal preference, that we only included completely randomized design studies or randomized complete block design studies, because the rationale to not include Latin square design studies was that Latin square design studies are relatively short period of time, and given the fact that amino acids they need to be supplemented for a given period of time, which is not like 2 or 3 weeks. It should be a little bit longer than that, which you can have in CRD or RCBD based design studies. So that's why we only chose those studies which are CRD or RCBD based designs and have a control group where no supplementation of lysine was a treatment group, and one group had rumen protected lysine group. So at the end of the day, we came down to 13 experiments with a data set over 600 dairy cows and 40 treatment means. And out of those 40 treatment means, 17 treatment means were without supplemental rumen protected lysine, and 23 treatment means were with rumen protected lysine.
I think, a lot of people on meta-analysis, they kind of skip over the materials and methods. But reading that inclusion criteria, I think, is critical. It finds out what your inference space is in your experiments. I think that's a really critical point, even if you're not a statistician, to read that section.
Exactly, because if you see now that every paper has two groups, one control, one rumen protected lysine, the experimental design is same. All papers supposed to present the feed ingredients, because some papers will be conducted using CNCPS model, some NASEM, some NRC. There is a huge variability in formulating diets for that experiment. So one of the idea was to collect the data of feed ingredients of each individual experiment and put those diets back into NASEM so that we can predict the metabolizable lysine content using the same model, so that all the studies are on the same scale. So that was also one of the reasons that we only chose those studies which provided the chemical composition and the feed ingredient composition of the diets.
Usman, we've done quite a bit of research with lysine, and one thing that we've learned is there's quite a bit of variability in the bioavailability of commercially available products. and they're not all characterized correctly. And so, how did you take that into account when reviewing your when putting this paper together?
It's a great point, Scott, because when it comes to the lysine, the encapsulation is not an easy job because there are a couple of things which are specifically associated with lysine. One of that is lysine is highly water soluble. So it takes to make an effort to make encapsulation of such products which are highly soluble in water. Second point is the lysine, the encapsulation of lysine, it varies when you mix the diets. So, for example, there was a study conducted a couple of years ago where they had 6 different products of lysine, and they performed the mixing of those diets. So, by the time those diets were mixed, 2 out of 6 products they were already releasing lysine. And the third point is it matters a lot the stability of the product within the TMR. So out of those 6 products, 3 products are already releasing lysine even before cows started consuming that product. So I guess the encapsulation technique, like Clay always says, all rumen-protected products are not same. The very famous sentence is correct. It's not an easy job. And the second point, as a scientist, I believe that we should test a rumen protected product well ahead of time before designing an experiment. We spend so much money, so much resources, and so much labor on conducting an experiment without knowing that if it would really show up in the blood or not. So I guess when we are designing the experiment, a pilot study should be designed on a cow level to see if this product is truly bioavailable or not. So another thing is you talked about bioavailability. So let me tell you this: if you go to the literature, the bioavailability of lysine products which are commercially available is from 5 to 80 percentage. So it's a huge variability. So and it matters a lot to account the bioavailability factor while formulating the diets, because you might be formulating it to match to the NASEM standards, but if the bioavailability is less, you are feeding less amount of lysine. So I think it's a major factor in all, not just lysine, in all those encapsulated products, that we should have a clear idea uh, of bioavailability. I would like to add one more comment here. There was a study which was conducted by Dr. Alex Haristo from Penn State University group. So what they did there, they established a criteria, a protocol, I believe, to measure the bioavailability, and then they compared 3 different lysine products which were available in market. So what they found is whatsoever lysine bioavailability was claimed by the manufacturer, they observed half of that. So, which tells you that yes, it is true that many of those bioavailability studies are conducted using in vitro studies, but I guess it is time now to test the bioavailability of those products in a pilot study using a cow model. So that's my take on this one.
So Usman, what values did you end up using in the meta-analysis and for bioavailabilities?
So I guess the range of bioavailability of the 7 products which were based in this meta-analysis was somewhere from 22 to somewhere close to 90 percentage. So the values which I used, if the authors reported those values in the paper, so I was bound to use those values because the diet was formulated according to that value. In case if the bioavailability was not provided, so then I contacted authors. Not every time you get a response back, and then eventually I contacted the manufacturing company to get to know what was the bioavailability and concentration of lysine in their product. So those were the values I used to formulate the diets.
Okay, thank you. Hey, Usman, I want to take a couple steps back here to earlier in the conversation. I think I'm aware of this, but maybe the others on here aren't. So congratulations on your new position.
Oh, thank you.
So tell us why you're in Switzerland.
So I finished my PhD from University of Florida, working with Dr. Jose Santos. Then I moved to University of Wisconsin-Madison, and I did little over 13 months of postdoc in Dr. Heather White's lab. Uh, I was also working closely with Francisco Penning-Garicano there. Francisco was also my committee member in my PhD program. But then I got to know this position in Switzerland. This is a position of research scientist and study director. So I applied for this position, and here I am working under the supervision of Dr. Muti and Neo. So the role is it's basically the role is like a faculty. So you get students. I already have 4 PhD students to supervise them. So the role is mentoring, teaching, and conducting research. So they name this portfolio as a study director. So that's where I landed up in
Oh,
Zurich.
congratulations.
Yeah, thank you.
Cheers.
Thank you.
And what university? I didn't hear. I might have missed it. Which university are you with?
It's ETH Zurich.
Okay. I'll eat some chocolate while you're there.
Yeah.
On this paper, we're going to talk of means a lot, but the difference in between control and I know there's variation. There, control and treatment was 7% of the MP is metabolizable lysine in control and 7.6 for the treatments. Is that a good reasonable range, or is that excessive? Or
So if you see the variability between control and treatment, yes, you are correct. So the lysine supplemented diets, they had 7.6 percentage of metabolizable lysine as a percentage of MP. But if you see the diets with rumen-protected lysine group, the variability was from 5.5 to 4.7 to 5.7 to all the way to 9 percentage 9 percentage of metabolizable lysine as a percentage of MP. So I would say the majority of the data, it existed somewhere between 6.5 and 8.5. And that's why those were the 2 values which I chose to predict response variables, because we wanted to predict values where most of the data relies and not predicting outside the range of the data limit. So what I could tell you, I did not find many studies with the very high concentrations of lysine. So only one of 13 studies, they supplemented close to 9 percentage of metabolizable lysine as a percent of MA, which tells me that not many studies were performed using high dose of lysine, which is a research gap to me to identify if we should supplement what more than whatsoever is recommended by NASEM.
you use NASEM and you got to use something, but do you think you'd have gotten similar overall results, say if you use Cornell or some other nutritional model? I don't know how the two models compare in predicting amino acids or MP, but you think similar results?
There is a recent paper which came out from Dr. Mike Vandenberg lab. They conducted a meta-analysis, and I guess they have a very large data set where they compared 3 different models, NRC, NASEM, and CNCPS. It came, I think, a couple months ago in 2024 in Journal of Data Science. According to that paper, the model predictions for the outflow of essential amino acids, the NRC and NASEM, they are very close enough. For some amino acids, CNCPS did not predict as NASEM or NRC were predicting, so it was below than their prediction. And for some amino acids, NASEM or NRC, they were over predicting some values. So I would say they are not equal. Because if you go to CNCPS, they recommend that 7% of metabolizable lysine as a percentage of MP is sufficient. But when it gets to NRC, the value gets to 7.2%. And when you get to the NASEM, the value gets close to 7.5 to 7.7 percentage of MP. So there is a variability depending upon what model you select to predict those values, I would say.
Okay. I guess we could go ahead and get into the results. We'll start with production. What kind of response do you, on that? We'll start with the average, and then we'll get into the regression type stuff. But on average, what was the difference in milk production?
So we conducted analysis in two different ways. So first of all, the analysis was done very straightforward. What happens when you supplement or you don't supplement rumen-protected lysine? So we only had the both treatments as a categorical variable. So in that scenario, the response is dependent upon the stage of lactation. Interestingly, in our data set, almost 40% of the data set was generated using early lactation cows, where rumen-protected lysine was supplemented uh, starting from 20 days in milk. And the half of the data set where rumen-protected lysine was supplemented during the mid-lactation. So interestingly, when you see in the data, the cows which were supplemented in early lactation, they produced 1.5 kilograms more milk as compared to control cows. But when you supplement rumen-protected lysine in mid-lactation, the response was still there, but the magnitude of response was less. So only rumen-protected lysine supplemented cows, they produced 0.8 kilograms more milk than controlled cows. So I would say uh, the milk yield in response to supplementation of lysine was increased, but it is dependent upon the stage of lactation. That was one of the factor. Another factor which we explored was the duration of rumen-protected lysine supplementation is also important. So the studies where people supplemented rumen-protected lysine 70 or less than 70 days, the response was less as compared to those studies where rumen-protected lysine was supplemented more than 70 days, and the difference was almost 50% less. So if you ask me in one sentence, studies where people supplemented rumen-protected lysine starting early lactation and for longer duration, they produced 1.5 kilograms more milk as compared to studies where people started supplementing during mid-lactation and for less duration, they produced close around 0.8 kilograms of more milk. So that it was one of the analyses that we did, what happened when you feed or you don't feed rumen-protected lysine. Another analysis which we did was we wanted to draw the relationship to see what happens to milk yield or other productive performance variables when you have lysine as a percentage of AMP. So in that case, we found a linear relationship. So increasing metabolizable lysine as a percentage of AMP increased linearly yields of milk, fat-corrected milk, protein yield, fat yield, or energy-corrected milk yield. So increasing metabolizable lysine from 6.5 to 8.5% of MP resulted in increase of roughly close about 2 kilograms more milk. But you never know what happens beyond 8.5 because there are no data points or there are no studies exist which can tell us that if there will be an additional benefit of supplementing beyond 8.5 or more than 9% of metabolizable lysine percentage of hemp. So that was, I think, the major results that we observed in this meta-analysis.
Do you think the bigger response in early lactation? This is you don't know the answer to this, but is it because they don't eat that much, or because they should be producing more milk than the later lactation, probably on average eating less? So which do you think it is, or is it just early lactation cows respond more?
I guess there can be several factors which can be discussed and taken into consideration. So, for example, during transition period, cows are not only experience net energy balance, they also experience net protein balance. If I recall data from Dr. Bell's paper, so he mentioned that transition cows experience almost 1 kilogram of protein loss during that period from skeletal muscle. So that's a huge amount of protein which early lactation cows they do that. Another factor is what Dr. Lourdes group from Illinois they pointed out. So what they did is so they measured concentrations of different amino acids during the transition period. So what they found is, so let me talk about just two amino acids, lysine and methionine. So they found that concentration of lysine in plasma, it starts going down starting 21 days before calving, and at day one or 1 day after calving, it reached at the nether point where the smallest concentration of lysine was observed in plasma. But that concentration came bounced back at 7 days in milk. But in case of methionine, so methionine also started to go down, and as calving approaches, and the smallest point was observed at 10 days after calving, which they gained back on 28 days, uh, 28 days in milk. So suggesting that, so the lysine, the utilization of lysine by the mammary gland or other tissues is extensive during the prepartum period, let's say 2 to 3 weeks before calving, and later on it's less. On the other hand, methionine, the use of methionine by the body tissues is probably more in right after calving. So like you said, besides these factors, yes, I agree with you that dry matter intake is less, and cows are mobilizing lots of protein. So if you supply amino acids or those nutrients which are going to be utilized by the mammary gland, the response will be way better. Another reason is the lysine, it is not extracted by the hepatic tissue. Majority of the lysine is extracted by the mammary gland, and mammary gland, the uptake of lysine is way greater than the output of lysine in milk protein. So I believe the lysine is not only utilized for the synthesis of milk protein, but it also utilized for the synthesis of branched chain amino acids or other non-essential amino acids. So if you supplement lysine before calving and during the early lactation period, so I would say the response would be, and the utilization to improve the efficiency for the synthesis of protein will be way greater as compared during the mid-lactation cows.
I think we can learn a lot by outliers. And I looked at one of your graphs, and if I did my counting correctly, just about a fourth of the studies in milk production didn't respond, or they were even a little bit negative. Did you look at why those four? I had 7 studies, but why those studies? Why the cows in those studies didn't respond? Did you try and find out, maybe to identify when you're more likely to get responses to these to RP lysine?
I'm not sure, Dr. Wise, if I get your answer question.
Okay. Well, like I said, if you look at some of your graphs, 7 studies or 7 comparisons did not respond positively. And did you look at to why those most of 3/4 of them were positive of the comparisons, but about 1/4 was not? Did you look at what might have been different between those 25% of studies where the cows didn't respond?
I guess one of the possibility could be the experimental unit used in those studies. So out of 13 experiments, only 4 experiments they had treatment groups where the experimental unit in their individual studies was more than 15. So 70% of the data which is generated from the individual study does not have 15 or more than 15 experimental units. So I guess one thing, the way I think one of the reasons we do meta-analysis is to integrate findings across all the data points to see the overall response. So one factor could be that majority of the experiments were not conducted by having lots of cows. So probably those studies were underpowered. That could be one of the things. The second thing, which Scott pointed out earlier, the bioavailability. So I guess it is possible that the bioavailability of the rumen-protected products was not enough to elicit the response as it is expected. So this could be the second factor, I would say. That's why you see the 25% of the studies, the comparisons, you don't see the response there.
you didn't look at maybe some unique diet aspects. And some of these are even to the left of the line, which wouldn't be power related. They've responded less. But you didn't look at, is there something unique on those diets or things like that?
No, no,
Okay.
I didn't look into that, but it's a good point.
Okay. Yes, the next thing is always interested in energy, and you got increased feed efficiency with RP lysine.
Yes.
More milk and about the same intake. So, what do you think is going on here? Suggests they're getting more energy per unit of feed. What do you think is going on there with that?
So we found an interaction between metabolizable lysine and metabolizable methionine for feed efficiency. So if you see the plots in those in the paper where we have where we plotted metabolizable methionine on x-axis, and we have those 5 lines with different levels of metabolizable lysine, those lines will tell you that whenever the diets were fed with 8 or 8.5 percentage of metabolizable lysine and 2.5 to 2.6 percentage of metabolizable methionine, the efficiency was greater. In other words, those diets, when the methionine level was less, the increased supplement the level of metabolizable lysine, the magnitude in the response to feed efficiency was greater when there was an increased methionine in the diet as well as lysine in the diet. So I guess it is a cumulative effect which is increasing the efficiency, overall efficiency. So it means that not only lysine but methionine, which is also an essential amino acid, they both are involved in improving the efficiency. And those two amino acids, as we know that the diets of US dairy cows, they're mostly on corn and soy based diets. So it matters a lot when you balance those amino acids which are critical and essential. So that is why you can improve the efficiency. Another aspect is the immune function, which is linked to those amino acids. So, like I said, methionine or lysine, they are the precursors for the synthesis of different antioxidants. Let's say like glutathione or some other ones which are necessarily needed to reduce the reactive oxygen species. So, in order to understand the link between immune function and amino acid, in this meta-analysis, I guess half of the papers they also presented data on somatic cell score. So, what I observed is the cows which had increased supplementation of lysine, they had less somatic cell count in the milk, suggesting that those amino acids they also improve the health of mammary gland. And there is some evidence in human literature and in monogastrics that those amino acids are also involved in the proliferation of cells, which tells me that probably the supplementation of amino acids, especially well ahead of calving, they can mobilize. They can not only synthesize the milk components, but also they might proliferate some epithelial cells, which are necessarily required to increase the synthesis of milk. So I guess those points might be involved in improving the overall efficiency of those cows.
So, Usman, from a feed efficiency perspective, depending on how much metabolizable methionine is in the diet. So, as feed efficiency improves, with lysine supplementation on the higher methionine diet, does that mean that we're seeing a higher milk response to the lysine if we have adequate methionine in the diet then?
So I guess if you have adequate methionine, then the response to supplemental lysine will be there, but the magnitude of that response will be smaller, I would say. That's my take on that.
Okay.
Because the overall intake, dark matter intake, wasn't related to in your regression to MP lysine content. Was there an interaction with methionine with intake and
No.
lysine? Okay, so it had to be just more milk then, not the efficiency had to
Right.
be driven by milk,
Right.
not intake. So
Yeah.
okay, I
So
was in. Oh, go ahead, Clay. Go ahead.
so I'm curious, sort of a little bit on that topic still. So I don't know if you looked at this value in NASEM or not. I'm curious, what's the lysine to ME ratio on these diets? Do you know what the range was for that metric?
No, it's no. I don't remember now. What was the range?
I, I'd be curious to know that at some point
Yeah, I will
because
send you an email on that because I have
yeah,
to go back to those NASEM sheets and look into that. But it's really top of my head.
yeah. No, thanks. I noticed it wasn't reported. I was curious though, because that is a metric we look at a lot in these diets
Yeah.
commercially. Yeah.
Yes. What surprised me a little bit, and I'm not an amino acid guy, milk protein was related to lysine as you'd expect, but so was milk fat percent, not yield. Yield was too, but milk fat percent increased with increasing MP lysine. What do you have an idea on the mode of action? Or they said I don't know what lysine does on fat synthesis, but it increased it in this meta-analysis.
Yeah, I really don't know what could have been there because I tried to look for some potential pathways and mechanisms which can be part of that. I found some information that lysine is a ketogenic amino acid and it can be associated with the synthesis of some ketones, but I really don't know what's going on there with reference to the milk fat synthesis.
I don't know either. I don't know if these guys know, but I don't.
So I would say commercially we do observe that a number of times. You know, that increase in milk fat percentage when we're supplementing lysine. There may be a mechanism there, I think, related to carnosine, perhaps.
So
Um,
yeah, well, related to carnitine, I know that lysine, if it gets a methyl donor, if it gets a methyl group from acetinosyl methionine, it converts into trans methyl lysine, and which eventually is used for the synthesis of carnitine. And now carnitine can be used for the beta oxidation in the hepatocytes. But I really don't know what would be the role of carnitine at the level of mammary gland, which can be used as a pathway for the synthesis of milk fat. But yeah, it's a good point to explore.
The response on milk fat was actually greater as a percent of the mean milk fat than the protein. I mean, it was a huge response on milk fat percent. I think the slope was 0.5, so really big.
Yeah.
I guess one question, on NASEM, we kind of got away from ratios. Not in NRC there was the lysine methionine ratio, but in NASEM, it pretty much was not discussed at all. Do you think there is an optimal ratio between lysine and methionine?
Well, I would say if you have a ratio between lysine 3 to 1, it should be enough. But like you said, I couldn't find also the ratio in NASEM. They only focus on now balancing the amino acids and not on the ratio.
I guess one more question on NASEM. Did your response to, you'd have to convert this to grams of metabolizable lysine, not proportion of MP. But did you look at the relationship between change in grams of metabolizable lysine to milk protein and how that slope compared to NASEM slope? I'm just
No,
curious.
yeah, no, I didn't do it, but it's a good point. I should do that analysis.
I'd just be curious to see how it fits. Hopefully it does, but who
Yeah,
knows?
no, I didn't do it. I guess
So
I guess sorry. When I was planning to do this analysis, there were a couple options. One was to do the relationship between metabolizable lysine as grams per day or metabolizable lysine as percentage of MP. I guess the reason we chose metabolizable protein as a percentage of MP because it's a function of dry matter intake, right? So in order to avoid collinearity, we chose to use that factor as opposed to use the grams per day. But but I would like to see that relationship as well.
Now, it makes sense what you did because intake drives everything. So you never know, is it an intake, a nutrient response or a lysine response? So I don't disagree with the way you did it. I guess one last question for me on the duration. Greater duration. Do you think this is just, again, because on protein studies, the response just takes a long time to get maximum response? Or do you think it's something else? And you started this by saying you don't like Latin squares for protein experiments. I agree 100% with your statement. But do you think it's just a time effect, or just it takes that long for cows to fully respond to any protein treatment, basically, whether it's lysine or methionine or just crude protein?
Yeah, I guess no. I think I agree with you that duration matters a lot because proteins or amino acids, they are not something which a body can store a lot for the needs of the, let's say, for the milk synthesis. So I get it. It takes a while when you supplement those amino acids and they get part of the body accreted into tissues, and then they start mobilizing and get into the play a role in the synthesis of milk or milk protein. So I don't have any other reason other than that it takes a while to see the effect of amino acids.
Yeah, kind of one last question for me, Usman, is that you've had an opportunity to look at multiple different studies and kind of dissect them and look at them. If you were to design the next lysine research, what would that look like? And this is a two-part question. Then the second part would be, do you anticipate that you'll be doing any kind of amino acid research?
So yeah, back to your first question. So I would let me give you an example from one of the meta-analysis that Jose did, I think, 3 years ago. So, in that meta analysis, what they did, they wanted to see what is the effect of metabolizable protein prepartum on the postpartum performance in primiparous and multiparous cows. So, in that meta analysis, what they found is between the range of the data set that cows, when they were fed 800 to 1100 grams of metabolizable protein in the prepartum period, the milk yield was response was linear. And if you just get the percentage of lysine based upon that meta-analysis, so that percentage was 8.2 basically. So the 8.2 percentage of metabolizable lysine as a percent of MP is an optimum one during the prepartum period to improve the performance of those cows. And based upon the results of this meta analysis, where you have a linearity until 8.5 or close to 9, I would like to design a study with increasing levels of metabolizable lysine as a percentage of MP, starting from the one which are recommended nowadays from with NASEM. So let's say one group is at 7.7, another one is at 8, and the third one is let's say at 8.5, or probably fourth one at 9. So I guess the finding from this meta-analysis was that only 4 studies out of 13 they presented in what happens in the primiparous cows. So we really don't know if those responses which are observed here would be also valid for the primiparous cows, especially when those cows are uh, accreting more tissues towards the mammary gland or towards their growth. So I guess there is gap to do studies with primiparous cows, and also there is gap to do studies with more dose titration experiments. The second thing, I did my postdoctoral work with Dr. Heather White, majorly focusing on rumen-protected lysine. And we not only focused with different levels of lysine, but also we evaluated the immune function of lysine. So basically, related to the immune function, we should be submitting that data soon, but we wanted to see what happens to the bioenergetic measures, specifically of T lymphocytes, when you supplement lysine or you don't supplement lysine. So yes, I conducted already 2 experiments with rumen-protected lysine, and in future, I hope so, I will be working with rumen-protected histidine, rumen-protected arginine, or some other amino acid work.
Very well.
So Usman, back, just talking about supplementation levels that were in these experiments. So, and maybe hitting on a few points that Bill had brought up earlier. Was blood meal used in any of the diets that were used in the meta-analysis, either in the controls or in the lysine supplemented diet?
I have to look carefully again, but as if I remember correctly, I don't see that if I added a paper in this meta-analysis which used blood meal specifically. But I will need to look again, double check this.
So the mean lysine supplemental lysine level was 19.3 grams of metabolizable lysine, correct?
Yes. I think the range was somewhere
So
from 5 to 40, but the mean was close to 20. Yes.
yeah, so Bill had asked the question earlier. So that mean level, that's a pretty good difference from control. That level of additional MP lysine. Of course, that 5-gram dose, that's a really minimal
Yeah.
dose, I would say, commercially. That 40-gram dose is a huge dose commercially.
Yeah, you are right. But I guess it matters a lot because, so for example, in Switzerland, uh, they have mainly forage-based diet. If you talk about US, they have mainly soy and corn-based diet. So I think that the ingredients of the diet matters a lot if you have to use 10 grams versus 20 versus 30 grams of metabolizable lysine. So I guess at the end of the day, it really matters what are the feed ingredients and what is the bioavailability of the source of lysine, which has to be used for the proper formulation of those amino acids.
Yeah. So there have been other lysine meta-analyses, conducted before. What were the basic differences between this meta-analysis and previous ones that were done?
So let me tell you one thing specifically. So the one meta-analysis which was done back in 2010 by Dr. Robinson, at that time we only had 7 studies, so very few number of studies with rumen-protected lysine. But the issue with that meta-analysis was, on the record, I'm not discrediting their work. I'm just pointing out what were the limitations. So basically, in their meta-analysis, they included all the studies which had rumen-protected lysine, or where the lysine was infused in the abomasum, or where the lysine was directly infused in the intestine. So basically, they did not segregate the studies with only rumen-protected lysine or with the lysine which was supplemented at the level of intestine or abomasum. So that was, I think, one of the major difference between that meta-analysis and this meta-analysis is that we only included those studies with rumen-protected lysine product and not included those studies where you infused uh, lysine at the abomasum or intestine level. I think that was one of the major difference. Another meta-analysis which was done by Dr. Irdeman from Maryland. I guess in that meta-analysis, they also did not differentiate between rumen protected or if the lysine was infused at the abomasum or intestine level. but also they only reported data on the milk protein yield. They did not report data on the intake or milk yield or milk components or amino acids or anything. The only response variable was milk protein yield, which was evaluated in that meta-analysis. So I guess uh, uh, if you talk about the differences, this meta-analysis included all the studies whichsoever available in the literature with rumen-protected lysine, and also covered majority of the response variables as compared to those ones.
I got one last question. I'm looking at my notes and I missed this. In your regression analysis, where you looked at the effect of milk or protein or whatever on change in MP lysine, did those data points include the controls? In other words, there was a lot of variation in the MP, the lysine as a percent of MP in control, or did it just include the treatment ones in that regression?
No, those regression points included all the 40 data points,
Okay.
all 40
Okay.
treatments.
Okay.
Yes.
Okay.
All right. Sounds like a good time to call last call then. If you guys will just, as we close this out, kind of give us a, 1, 2, 3 kind of key takeaways that you guys take away from this research. And Clay, why don't you start us off?
New research is changing everything we thought we knew about choline's impact on the cow and her calf, and top scientists have a lot to say about it. They are presenting new research that supports choline as a required nutrient to optimize milk production, choline as a required nutrient to support a healthy transition, choline as a required nutrient to improve calf health and growth, and choline as a required nutrient to increase colostrum quantity. This new research is solidifying choline's role as a required nutrient for essentially every cow, regardless of health status, milk production level, or body condition score. Learn more about the science that is changing the game and the choline source that is making it happen. Reassure precision-release choline from Balchem. Visit balchem.com/scientistsay to learn more.
Yeah. So I was very excited to see this meta-analysis. Obviously, we have a lot of interest in amino acids, lysine and methionine. So I think this really, this validated a lot of the things that we've talked about for a number of years. That the earlier in lactation you start supplementation, and the more likely you are to see a response, and you're not so likely to see a response to the lysine if you're methionine deficient as well, right? So it's important to really co-supplement these amino acids. So I think there's a lot of good practical takeaways here that we can utilize in the field. And I would be curious, Usman, as a follow up, what those lysine to ME ratios are, because I'm very interested in seeing how that metric would compare as well. But thank you, very very nice paper.
Yeah, great paper. Bill, any final comments from you?
Well, I like, you excluded a lot of data, but for good reason. And I think that makes the actual conclusions more applicable. Like, you got rid of infusion data and all this, and it makes it much more applicable. And again, I urge people who read meta-analysis to read exclusion data. And the other thing I like about this is the regression part, the quantifying the response. It again helps people determine, is this going to be economical? What's the expected response? So those two things, I think, make this a really good paper.
Yeah. And Dr. Arshad,
Well,
any final comments from yourself?
Thank you for the appreciation. I guess there is no question about that cow needs a balanced ration, which also includes balancing not only essential amino acids, but overall amino acids. And also, it is very important from research perspective that we should take account the bioavailability component strictly, and we should do some pilot studies to confirm the bioavailability of the product before we start utilizing the resources. And the third thing is, I would say there is still need to identify the optimum dose of lysine for the performance in cows. And we still have a research gap to understand the role of supplemental lysine in primiparous cows, because majority of the herds, they have 30 to 35% of the cows which are primiparous. So it will be very important to understand the role of supplemental amino acids in the primiparous cows, which contribute 30% of the herd dynamics.
Thank you. Well, I got to say, this has been very enjoyable. It's been a great way to spend an evening. So I want to thank you guys for your time and your knowledge and spending some time with us here this evening. To our loyal listeners, as always, we thank you for coming along with us as well. We hope you learned something. We hope you had some fun. We hope to see you next time here at the Real Science Exchange, where it's always happy hour and you're always among friends.
We'd love to hear your comments or ideas for topics and guests, so please reach out via email at anh.marketing@balchem.com with any suggestions, and we'll work hard to add them to the schedule. Don't forget to leave a 5-star rating on your way out. You can request your Real Science Exchange t-shirt in just a few easy steps. Just like or subscribe to the Real Science Exchange and send us a screenshot along with your address and t-shirt size to anh.marketing@balchem.com. Balchem's Real Science Lecture Series of webinars takes place on the first Tuesday of every month, with the top research and nutrition topics that will impact your business. We also include small ruminant, monogastric, and companion animal-focused topics throughout the year. Visit balchem.com/realscience to see the upcoming topics and to register for future webinars. You can also access past webinars and search for the topics most important to you.